So, Professor Tavernarakis, we have, not by coincidence, we have the molecular link between aging and neurodegeneration. So, Professor Tavernarakis is chair of AIT Governing Board, which is European Institute of Innovation and Technologies. He is based here in Heraklion in Crete. He is home. So, we'll see now in terms of research, in terms of aging, to prove the theory that you have to carefully choose the parents.
Thank you, Florian. Thank you, everyone, very much for being here this morning. It is a pleasure for me to again participate in this exciting conference once more. Thank you for the invitation, especially since this is happening now in my own hometown, home country here in Greece and in Crete. So for the next few minutes, I'm going to be talking about some work that we are doing in the lab, linking the process of aging with neurodegeneration, which, as you know, is a condition that sometimes is associated with aging. So let me begin by giving some pretext, some context in the work that we are doing. As you know, lifespan is increasing, human lifespan is increasing, especially in the last decades, as you can see here. The segment of the people above 85 are the fastest growing segment in the population right now in countries, for example, like Sweden and Japan, where we have data. As you can see, the growth of this segment is even faster than exponential. And this, of course, creates problems because aging does not come along, does not just arrive, but also is associated with several pathologies, some of them devastating, such as neurodegenerative disorders, cardiovascular disorders, diabetes, cancer, stroke, etc. All these pathologies are now becoming more and more frequent because the human population is aging. These used to be rare conditions, especially neurodegenerative conditions like Alzheimer's disease, but now they are more and more frequent. So what can we do to really increase the quality of life? As Professor Marcus said, you don't want to live longer if you are in pain, if you are suffering from these diseases. So how can we learn about aging so that we can battle these diseases that are associated with aging? This is what motivates our research. And of course, we are trying to understand aging by studying cells, specific cells, that are long-lived. And as you may know, neurons in our bodies are the most long-lived cells. Some of these cells are born even before we were born and will literally die after we are dead. So these cells, they are non-dividing cells. They will survive for tens or even more than 100 years, in the case of Professor Marcus, for example. So we want to learn from these cells. How do they make it? How do they survive for this long? So that perhaps by identifying the mechanisms that these cells use to survive this long, we could use these mechanisms to intervene in the case of pathologies associated with aging. Now, these cells, in addition to living long, they also face numerous challenges. They are highly energy-dependent cells, so they need a lot of energy to function, as you know. Even just sitting there, neurons, even just idling, they will consume a lot of energy just to maintain their plasma membrane potential. They are highly differentiated cells with complex morphology. They need to move stuff around. Some of our neurons have appendages, axons, that are more than 1 meter long. And you can imagine what challenges this poses to trafficking materials, proteins, back and forth from remote appendages like synapses, dendrites, back to the soma. So they have this unique architecture. And of course, because of that, because they need a lot of energy to function, they depend on mitochondria. Mitochondria are energy-producing organelles in our bodies. Actually, you saw already, the respiratory chain is there that produces ATP. And these organelles need to function perfectly for the duration of the lifespan of these neurons. Otherwise, cells will die. And exactly this is happening in cases of pathology. what we see, for example, in Alzheimer's disease neurons is that they accumulate damaged and dysfunctional mitochondria. This is a feature, a very characteristic feature of neurodegenerative diseases, this accumulation of damaged mitochondria. Now, we don't see this accumulation of damaged mitochondria just in the context of neurodegenerative diseases, but it appears to be a common denominator of many diseases associated with aging. And this is just a partial list of some of these diseases shown here that show this commonality, this common feature of damaged mitochondrial accumulation. But even just in the context of normal aging, we see this accumulation of damaged mitochondria. So, for example, if we compare cells in rats, where this experiment has taken place, 6 months versus 24 months of age, again, there is accumulation of damaged mitochondria, as you can see here. And not just in rats, even in other organisms remotely associated with mammals. For example, C. elegans is a Sinorhabditis elegans is a model organism that is used around the globe for studying neurodegenerative diseases, cell death mechanisms, and aging. Again, we see this gradual accumulation of mitochondria. Now, what intrigued us to go into this line of research was an initial observation that was made in a lab some years ago by a talented postdoc. What Marta actually observed is that while in wild-type animals we see this accumulation of damaged mitochondria, in long-lived mutants, in mutants of C. elegans that live longer than normal, we don't see this accumulation. And there are such mutants in C. elegans that are extremely long-lived. And those mutants have, for example, compromised insulin IGF-1 signaling or TGF-beta signaling. And these are just two examples shown here. And actually, if we go and intervene and alter the rate of accumulation of damaged mitochondria, make it either accelerated or decelerated, then we can even alter the lifespan of these animals. So whenever we intervene to accelerate mitochondrial accumulation during aging, we actually shorten the lifespan of these animals. Whereas if we intervene to decelerate, to ameliorate mitochondrial accumulation, we dramatically extend lifespan of even long-lived mutants. This suggests that there is a causative relationship between the mitochondrial accumulation and lifespan. Actually, enhancing this accumulation shortens lifespan, whereas blocking it can extend lifespan, at least in model organisms. And using this knowledge, maybe we can translate it to humans. But before we do that, we need to delve more into the mechanisms that bring this change about, that bring this gradual accumulation of mitochondria about during aging. And working with model organisms such as C. elegans and the mouse, we perform genetic screens to identify genes that are involved in the process of regulating mitochondrial content in eukaryotic cells. And I'm not going to go into the details for the sake of time, but I'm going to just focus on 3 genes that we recovered out of this screen. These genes are shown here, DCT1, NIX, that's one gene, PINK1, and Parking. These are 3 genes that are involved in a process called mitophagy. Now, mitophagy, for the aficionados, biology aficionados among you, is a type of cargo-specific autophagy, a self-degrading process that cells actually have to cleanse themselves of damaged components. And in this case, mitophagy targets damaged mitochondria, or it can even degrade normal mitochondria in a physiological setting. For example, our red blood cells are devoid of mitochondria. In addition to not having a nucleus, they don't have mitochondria. Very wisely so, because red blood cells transfer oxygen, and you don't want mitochondria that consume oxygen to consume the very cargo that these cells need to transport to our tissues throughout our bodies. So mitochondria are eliminated through a process of mitophagy during maturation of erythrocytes to red blood cells. And that's a physiological paradigm of mitophagy. But there are also pathological paradigms of mitophagy. For example, in the case where a dysfunctional, a damaged mitochondrion is detected inside the cell, then this process is triggered through the specific activation of the so-called pink parking pathway. Now, pink is a serine threonine kinase that phosphorylates, it's an enzyme that phosphorylates parking, which is another enzyme, an E3 ubiquitin ligase, and this ubiquitinates, attaches ubiquitins to specific proteins on the outer membrane of mitochondria. And this is the signal that suggests that this mitochondrion is damaged and needs to be degraded. And that's how mitophagy works. Now, in C. elegans and in other models where we studied mitophagy, we actually, as I said, identified these three genes that are arranged in this linear manner. So pink one activates parking, which activates DCT1, and that triggers mitophagy. And if this process is triggered, then we have lifespan extension. If, on the other hand, we inhibit the activation of mitophagy, for example, under conditions of either dietary restriction, starvation, or under low insulin signaling, for example, in mutants with compromised insulin signaling, then we shorten lifespan. This suggests that mitophagy activation, mitophagy is a process that contributes to lifespan extension. And indeed, it becomes activated under stress. It's a stress response mechanism that our cells have to fight damage that is accumulating inside them. And this is something that we can measure in vivo now, having identified specific genes that are regulating this process. Now, if mitophagy is not functional, if mitophagy is not happening, as you can imagine, then we have accumulation of mitochondria throughout cells and tissues, especially neurons. Now, this creates a problem for neurons because accumulation of damaged mitochondria results in oxidative stress being imposed on these cells. Why? Because damaged mitochondria, in contrast to healthy mitochondria, produce much less ATP and a lot of reactive oxygen species, ROS, which damages proteins, lipids, DNA inside cells. And the most proximal target of these reactive oxygen species, as you can imagine, is mitochondria themselves. So essentially, not being able to perform mitophagy results in an accelerated aging of mitochondria. Mitochondria actually will sustain damage during their lifespan, but this will happen at a relatively low pace. If mitophagy is not there to eliminate damaged mitochondria, then this accelerated accumulation of mitochondria will result in oxidative stress for these neurons. Now, neurons have ways to sense oxidative stress and actually cope with this onslaught of the oxidative stress. How can they do that? If neurons sense oxidative stress, what they actually start doing is transferring damaged mitochondria back to the soma for degradation. Neurons are, as I said, highly differentiated cells with very long axons, in most cases, very long appendages. And these appendages are filled with mitochondria, functional mitochondria, but if these mitochondria become damaged because of oxidative stress, then they are transferred back to the soma for degradation. And this is a defense mechanism of these neurons to defend against oxidative stress. This process of retrograde trafficking of mitochondria is triggered by specific signals, such as calcium, that is leaking out of damaged mitochondria. It's emanating out of damaged mitochondria. This is the signal that cells perceive to detect damaged mitochondria inside them. This increase in calcium activates calcium-dependent calmodulin kinases, essentially a kinase cascade that will result in the retrograde trafficking, will activate retrograde trafficking of mitochondria. So this is the mechanism by which mitochondria are trafficked back to the soma, where mitophagy is actually stimulated to degrade the damaged mitochondria. And that's how neurons cleanse themselves of the damaged mitochondria and can survive for very long. Now, if mitophagy is inhibited, if we inhibit, we intervene and inhibit mitophagy, we block it, then these neurons cannot cleanse themselves anymore, cannot really eliminate damage. So damage becomes accumulated, and this has devastating consequences. For example, it now renders neurons vulnerable to neurodegeneration. In this case, we can actually visualize this increased vulnerability by overexpressing, creating a C. elegans. In this case, we use C. elegans as a model, a Parkinson's disease model in C. elegans. How? By overexpressing human alpha-synuclein. As you may know, alpha-synuclein is a protein that has been associated with Parkinson's disease pathophysiology. So it becomes accumulated in Parkinson's disease patient neurons. Same thing with C. elegans. If we overexpress human alpha-synuclein in the dopaminergic neurons of C. elegans, because that's the kind of neurons that degenerate in the brains of dopaminergic of Parkinson's disease patients, we see the exact same thing. There is neurodegeneration, but this neurodegeneration is exacerbated to a great extent if we downregulate mitophagy. We essentially increase the sensitivity of these neurons to neurodegenerative insults by not allowing them to perform mitophagy and cleanse themselves of damaged mitochondria. In another model of neurodegenerative disease, Alzheimer's disease, here, as you may know, two proteins have been implicated, beta amyloid and tau. These are two proteins that aggregate in Alzheimer's disease patients. If these proteins are allowed to aggregate, they very strongly inhibit mitophagy, both in model systems, such as C. elegans, but also in the mouse, and in humans. We have been able to examine post-mortem brains of Alzheimer's disease patients, and we see that mitophagy is strongly inhibited in the neurons of these patients, suggesting that mitophagy is actually a process that protects neurons, and if it becomes blocked, then these neurons are vulnerable to neurodegeneration. Now, in addition to a pathological setting, mitophagy is also very important in performing essential physiological functions by neurons, such as, for example, learning and memory. In this case, this is an associative learning paradigm where we train animals to recognize specific scents if we pair them with a condition, for example, starvation. Starvation is a punishing condition. So animals that have been trained by pairing a specific chemical to starvation, they now learn not to like this chemical. So as you can see, chemotaxis in this case goes negative, suggesting aversion towards the chemical. This indicates behavioral plasticity, which is a sign of learning and memory, of synaptic plasticity and learning and memory capacity. Now, if we inhibit mitophagy, if we don't allow mitophagy to proceed, then these animals, although they do behave some behavioral plasticity, they never learn to avoid these chemicals, suggesting that there is a defect in physiological functions such as learning and memory in these animals. And we can actually restore such learning and memory deficits that are associated with dementia conditions such as Alzheimer's disease, as you know, by restoring mitophagy, by upregulating mitophagy using compounds that are specific activators of mitophagy. In this case, urolithin A is a natural compound that has been found to specifically activate mitophagy. And you can see that we can restore learning and memory capacity in these animals that are overexpressing beta amyloid and tau, two proteins that have been associated with Alzheimer's disease. So just to summarize, and this is my concluding slide, what I've told you today is that neurons are very long-lived cells, which indicates that they have increased powers of homeostasis. They can repair damage that is incurring during aging in these cells. And what is the mechanism, specifically with regard to energy production in mitochondrial homeostasis? The mechanism is mitophagy. Mitophagy is a specific type of autophagy that protects neurons from the accumulation of damaged mitochondria. If damaged mitochondria are sensed inside these neurons, then very quickly neurons eliminate these damaged mitochondria by transporting them to the soma, activating mitophagy to eliminate and recycle the damaged mitochondria. And this prevents neuronal degeneration, neuronal cell death. However, if mitophagy is not there, if mitophagy is inhibited, then These neurons are defenseless against the accumulation of damage and eventually will degenerate. On the other hand, activation of mitophagy can actually render these neurons more resistant to the accumulation of damage, and this can result in organismal lifespan extension. This is something that we have shown experimentally, both in simple models, such as C. elegans, but also in mammals, such as in the mouse. If we activate mitophagy further, then we extend the lifespan of these animals. And I will close by thanking my friends and colleagues at the University of Crete. This is nearby here, just 10 kilometers away. And especially Kostas and Irini, the two people that did most of the work that I presented to you today. Thank you very much for your attention, and of course I would be happy to take any questions that you may have.
Any questions? Just as an exception, normally we don't do questions, but since you are coming for the plenary, since you are coming from Professor Marcus, we can have one or two questions. We are a little bit late, but that's okay. Yeah, photo there.
Hi, thank you very much for the excellent presentation. I have a few questions because we're in the same field. First off, regarding mitophagy, were you able to analyze local mitophagy deficits, for example, in synapses, synaptic deficiency versus neuronal cell body deficiency, and did you see any significant differences?
Yes, so we have developed tools. I didn't really elaborate much because of the time limitation, but we have tools to locally monitor mitophagy in synapses specifically, but also in other cellular compartments, not just synapses, dendrites, axons, etc. Yeah. So indeed, mitophagy can take place in synapses, but most of mitophagy is actually taking place in the soma. That is why you need to transport most of mitochondria back to the soma where the lysosomal system resides, and that's where degradation takes place. So we do have some activation of mitophagy at synapses, but the bulk of mitophagy is actually activated in the soma.
Perfect. Thank you very much.
We have another question. Quick.
I don't need a mic. What kind of therapies does the research suggest to be on the horizon for these kind of neural conditions?
Yes, so of course we do this research to understand the molecular mechanisms that cells use to defend against insults. We hope that this knowledge will eventually find its way into therapies and interventions. Now, as I said, there are specific chemicals, compounds, that can activate mitophagy. I gave you the example of urolithin A, which is one such compound, but it's not the only one. There is a number of chemical compounds that can activate mitophagy specifically. What we found is that these compounds can ameliorate neurodegeneration in model organisms and extend lifespan as well. So we believe that translating this knowledge to humans might have beneficial effects towards combating neurodegenerative conditions such as Alzheimer's. And we are now in collaboration with pharmaceutical companies in the United States and in Europe, and also with clinics, both here in Greece and in Europe, to start clinical trials based on these compounds. So it's still too early to speculate whether or not such interventions will be effective in humans, but I believe this is knowledge that can be eventually can become useful in treating such diseases.
Yes, we have another question. Very quick.
It brought me to another question, if I may. You explained how mitochondria had to be brought back to the soma for the lysosomal compartments to degrade it. But in exceptional cases, for instance, in the spinal cord, where we have axons that go all the way to 1 meter in length in humans, would there be specific lysosomal compartments in the synapses in those cases?
Yes, there are. There are, not just at synapses, but also along the axon. So actually, these processes are concurrent. Transport is concurrent with formation of autophagosomes and fusion with lysosomes. And this can happen both at synapses a certain extent, but also within axons, within the long axons.
And if we could go back to an interesting slide where you show the tau aggregation model versus the alpha-beta amyloid aggregation model. Did you find any significant differences in those two types of Alzheimer's for mitophagy?
So both these proteins, tau and beta amyloid, aggregate and can strongly inhibit mitophagy. So this is what I showed you.
Yeah.
Aggregation of these two proteins can inhibit mitophagy. Now,
Yeah.
there are, depending on the context, differences in Alzheimer's disease patients with regard to the accumulation of these two proteins. Some patients will show more tau accumulation, others more beta amyloid accumulation. Regardless of what accumulates, both can actually inhibit mitophagy very strongly, as you can see.
Yes.
So we believe that this is one of the reasons why these neurons degenerate, why Alzheimer's disease patients lose their neurons. Because mitophagy is inhibited, rendering these neurons defenseless against the accumulation of damaged mitochondria.
Does one have more significant impact than the other?
No, we haven't been able to detect any significant difference. As you can see, they are about the same extent.
All right.
So thank you. You can, Davis, you can speak with Nektarios later. I know
Thank you
you
very
are
much.
in the field. Thank you very much. I really appreciate. If you can stay for a picture, we normally stay for just the record, you know. Thank you also. I guess we have a kind of idea now when Professor Marcus said you have to choose your parents. Should we manipulate this to choose your parents?